Hi, I'm Brian Jacobson, one of the associate editors of Gastrointestinal Endoscopy. I'm here with Dr. Walter Park from Stanford University in California. We're here to discuss his paper, Metabolomic-Derived Novel Cyst Fluid Biomarkers for Pancreatic Cysts. Welcome, Walter.
Thank you, Brian.
Can you tell us first, what is metabolomics?
So metabolomics is the study of metabolites by disease association. So what we're trying to do is look at different metabolites and correlate to disease states. It's very similar to what genomics is when we look at one disease state and we look at whether there are differing DNA levels. And like proteomics, which is really the study of different protein levels by different disease states. And metabolomics really is just one further extension where we're now looking at beyond DNA, beyond RNA and proteins, we're actually looking at metabolites and to see if we can learn something about a disease, and in this case pancreatic cysts, by the different types and volumes of metabolites.
And so this is something you can measure in blood, but also in cyst fluid.
Yeah, traditionally it has been studied or attempted to be studied in blood, but the amount and sheer number of metabolites are voluminous and quite complex. So serum-based studies with metabolites have been pretty limited, despite their theoretical promise. So one of the things we thought of in this particular study was that the cyst fluid would theoretically is a protected cyst fluid space, and that there would be less metabolites so that it would be more feasible to do a metabolite study.
It's very innovative. So can you give us some examples of metabolites that you found and measured in these cysts?
Yeah, so in this study, we had been collecting, for the last several years, we have been banking pancreatic cyst fluid with the hopes of using novel technologies like this to better understand disease, pancreatic cysts in this case. And going into this, we really didn't know what we were going to find. And so this wasn't really hypothesis-driven. This was really kind of empirically driven. And we took 29 cyst types that were defined by histology. The vast majority of them had gone to surgery, so we were pretty sure what type of cysts we were dealing with. And then we performed a semi-targeted metabolite approach, and we found that we were able to detect 500 different metabolites. Then comparing that to the clinically relevant cyst categories, we found 10 that were expressed at different levels. Because this was an exploratory process, this could have just happened by chance. So we repeated the study with another set of 19, a validation cohort, and we found
19 known cysts. This
yes,
was known diagnoses.
Again, yes, another set of 19 separate cysts that had undergone surgery. So we were near 100% confident of what these cyst types were. We found that of the 10 that we had observed to be differentially expressed in the first cohort, 4 of them we reproduced again. We then used mass spectrometry technology to try to identify what these 4 were, and we were able to identify 2. The other 2, despite our best attempts, we couldn't give a name to the actual metabolite. But the 2 we did find included glucose and kynurenine.
Glucose and kynurenine. Okay.
Yes.
So is there any thought as to why glucose and kynurenine would be differentially expressed in, I'm assuming, mucinous type versus serous type cysts?
Yes. So to step back, we compared, when I talked about clinically relevant cyst categories, the first category we compared included benign cysts with no malignant potential, and that included pseudocysts and serous cystic adenomas, versus cysts with cancer potential, and that included the mucinous cystic neoplasm, the intraductal papillary mucinous neoplasm, also called IPMNs, and cysts with proven cancer. And what we observed in the benign versus the malignant potential, and that included those with malignancy, we observed different levels of glucose and kynurenine levels. We were surprised initially, one, because glucose is a molecule that we so commonly study and analyze on a daily basis. We're not certain as to the actual mechanism. At a broad level, we do suspect that cancer, consistent with cancer biology principles, cancer has a higher metabolic rate. So there may be increased glucose consumption of cancer cells, which may explain why we saw a difference.
So you saw lower glucose levels in the cyst with a pre-malignant potential.
Correct. Preliminarily, we've done some work to kind of define that mechanism, and we've observed glucose transporters to be upregulated in the premalignant cells. And so that might be one potential mechanism. But at this point, we're not certain.
So these don't sound like commercially available assays just yet, or are they available in all of our hospitals?
For glucose, when we observed that glucose was differentially expressed, the first thing we did was I got on the phone and called our director of our chemistry lab and asked if we could collaborate, and we sent the same samples down to our chemistry lab, and he was able to run the same samples, and we were able to quantitatively observe a difference. Now, the caveat being is that although these assays are commercially available, and in theory anyone could potentially do it now in their own hospital laboratories, these assays haven't been validated for this specific purpose, for cyst fluid. So the caution is there are studies that need to be done to further validate and define the technical parameters of how they would perform in cyst fluid. Kynurenine, there is no commercially available assay at this point in time. There are ELISA assays that are used for research, but nothing available clinically yet.
Well, this is all very exciting preliminary first steps, and I know we all look forward to seeing the next things that come out of your laboratory. Thanks, Walter.
Thanks, Brian.